Today's podcast is a bit different than some of our others, and it's also the first official podcast in a mini-series co-sponsored by the ASGCT Ethics Committee. After our recent long discussion with Ben Hurlbut on the ethical considerations around germline genetic editing, the chair of the ASGCT Ethics Committee, Thomas Wechsler, and I realized that there are a number of intersections between ethics and the work we do to develop novel gene and cell therapies. And as a result, a mini-series was born. Today, we're going to dive into two topics that teams bringing novel therapies in clinic for the first time face: selecting the starting dose for a therapy and determining the acceptable off-target and/or non-productive on-target editing profile prior to testing a therapy in the clinic. Now, to be sure, both of these topics are deeply rooted in technical and scientific considerations. However, because at present, AAV-based therapies are typically one and done, which means that redosing is not yet possible, there's also some deep ethical considerations as well. So we're going to explore both of these topics today from both dimensions with our guest, Thomas Wechsler. And now, on to the show. Welcome to The Issue on the ASGCT Podcast Network. Thomas Wechsler, so happy to have you here today. Before we dive into today's unique discussion, could you tell the audience a little bit about your background?
Yeah, thank you so much for having me, Emily. So my scientific background is in DNA repair and genomic instability. And initially in my career, I was able to leverage that to get into Sangamo Therapeutics back at Biosciences, which was one of the first companies that actually developed gene editing even before CRISPR BC. And I was with zinc finger nucleases. And at the time, we filed some INDs in this space, starting with CCR5 before my time. And then we had some safe harbor programs in albumin locus, cell therapy for beta thal and sickle cell disease as well. And afterwards, I helped a small and rare disease company called Gray Science LLC to develop gene therapy for ANGLA1 deficiency. And that's in clinical studies right now. And then after a short span consulting, I spent the last years at J&J IM, Innovative Medicine, which is the new name for Johnson & Johnson Innovative Medicine for Janssen Pharma, where I helped build an internal gene therapy discovery team until, unfortunately, that was just dissolved very recently. And what got me interested in ethics and gene editing was the real possibility of germline editing and the ethical issues associated with it, which was something you extensively discussed with Ben a couple of weeks ago. And at the time, I had read this book called Our Post-Human Future from Francis Fukuyama in the early 2000s. And it really struck me, the ethical challenges he laid out, and he proposed new international regulatory bodies and highlighted the, responsibilities of the scientists, which at the time was sort of us, right? Because we were the first ones working on this. So I got into the educational committee at ASGCT at the time. We brought in bioethicists to the meeting, Hank Greeley at the time, who made a really good point about the anxiety that comes with new technologies, it being early recombinant DNA, IVF, gene editing, now AI, and how society deals with it. That was very illuminating. And then also went to the 2015 Gene Editing Summit in DC in person, which was very, very cool, because around the same time, Sangamo actually went to the RAC meeting presenting the first in vivo genome editing. So it was an exciting time. And what struck me was, of course, that none of these closing statement guidelines really had an impact, what happened then in 2018. So it's a continuous struggle, I guess, to put these guidelines in place. So while all this is still very important, and I think Ben and many others work on this, I think the current work of the ASGCT Ethics Committee is to look at ethical questions that arise during end-to-end development for gene therapy and gene editing. And just to kind of a little plug here, over the next few months, we'll touch on genetic testing, hopefully in form of a symposium at our annual meeting in May. We will cover clinical trial designs. This podcast is part of that. We'll have an article on molecular therapy, hopefully, on assessing clinical failure and success. And then we'll also have some content how gene therapy competes with other modalities in the future. And yeah, this is the work of a great team. And please, look out for that.
Absolutely. No, that it's sort of perfect timing that you and I were able to come together and have this conversation. And, in preparation for it, we talked a little bit about like what are the two topics that I think pretty much every development team in this space has to face as they start to think about their project. Assuming they're working with one of the technologies that we're about to talk about, that is. And so, we're going to start and we're going to talk about the ethics and science of starting dose selection. And we're going to kind of maybe narrow our focus there and talk about it in the context of AAV therapies, because again, this podcast is meant to be, digestible in 40 minutes. We could keep going for hours if we explored all the kinds of cell and gene therapies that are out there. So we're going to keep it a little bit focused on starting dose selection in AAV. And then we're going to talk a little bit about the ethics and science of identifying acceptable off-target and undesirable on-target editing profiles when you're using different kinds of gene editing technologies. And so what's great about this and why I'm really excited to be here with you is that you and I actually worked together many moons ago while you were at Sangamo. I was on the Shire side of the Shire-Sangamo Alliance, which was one of those safe harbor programs. And so it's in some ways, I think, we both had a front row seat for some of the earliest forays into this. And on the one hand, I'm glad to see, how far the technology's come, and we'll talk about that in a second. But on the other hand, I think the principles that we struggled with are the same ones that teams struggle with today. So let's just dive in. And I apologize. I'm going to try very much to have this not be a monologue, but I feel like for starting dose selection, I got to do a little bit of a preamble so that our listeners sort of understand where we're coming from. So just a quick preamble is this. I started my career in small molecules, and small molecule starting dose selection for a dose escalation trial is pretty, dare I say it, straightforward. You do tox in two species, one rodent, one non-rodent. You do pharmacology and hopefully one of those two species as well. You calculate, your therapeutic window, which is, the difference between the dose where you're seeing, efficacy and the dose where you're seeing tox. Depending on the nature of the tox, as well, you'll maybe caveat some of your dose selection calculations, but there are like standard formulas for saying, if this is the dose where I'm seeing efficacy in the mouse, this is probably what it's likely to be when I extrapolate to a human with the human-sized body involved, right? And the difference here is that with small molecule drugs, if the starting dose is not the efficacious dose in the clinic, you get a second chance, right? Because if a patient's part of a first dose group and that dose group is below the level of efficacy, you can absolutely roll them over into another arm of the study where they receive the dose that was later determined to be efficacious during dose escalation. That's not possible with AAVs today. And the reason it's not possible is because at this point, we can't redose. And hopefully that'll change. But at this point, AAV therapies are considered one and done, which means number one, you only have to take it once. But number two, if the dose is wrong, if it's too low and it's not in the efficacious range, then that subject can't receive that same therapy today in order to bump up their levels and bring them to an efficacious level. And so let's talk maybe a little bit, Thomas, about what you've seen teams go through, and I'll probably jump in as well, because I think you and I have seen a lot of different teams sort of struggle through this. At the end of the day, the goal is about balancing the risk and benefit for each trial participant. And so how have you seen teams struggle through these deliberations?
Yeah, I think compared to some of the other modalities that you just referred to, like small molecule biologics, I think the starting point is usually very similar, right? You basically define a target product profile or concept where you sort of log in, okay, this is where I want to end up. For example, for hemophilia, you want to be somewhere between 5 to 100%, which is therapeutic. And you basically then
in factor levels, right? Yeah.
Yes, exactly. Afterwards, assuming again, in this case, you start something around 1% or even lower. So you go for like a normal sort of like drug development process. You have lead optimization. In this case, it's the AAV vector, which is both the capsid and the transgene. So you optimize both as well as you can do. And then, you experiment with the doses. And very similar to what you just described, you try to establish a minimal effective dose. And then you also escalate to something where you see a safety signal, which is either your maximum tolerable dose. In the case of AAV, sometimes you have a maximum feasible dose because you cannot dose higher. And then you basically try to go into like a small and a large model. You usually have a rodent model that's sort of your disease model, where you can sort of establish the PK-PD relationship. And then you go into a larger animal like an NHP for PK only, because these are usually wild-type animals. There's some, disease cases where you can induce the disease, but that's usually has its own ethical challenges. So the dose scaling itself, I think, is the first difference between small molecule and AAV, because we know that serotypes not always necessarily transduce all species the same way. So you have to sort of certain scaling factors in there. It's also not easy to just sort of escalate based on body weight from a mouse to a monkey, right? Because sometimes the organs in either species are completely different. So sometimes you use organ weight or sometimes even organ volume to scale from a mouse model to an NHP model. So these are all sort of like, complexities you have to consider. And then, of course, in the case of AAV, it's also that AAV itself, as you mentioned, is immunogenic. So it, triggers a pretty strong immune response you can control with steroids. And if you have certain comorbidities in some of these disease models, it can lead to additional complications. They may not be visible in a mouse model, and then you can just basically observe it in the clinic. And that happened at extremely high AAV doses, unfortunately, for some programs. And so these are some of the really big challenges, which these programs usually address fairly late in the program. So I think that there was definitely a bit of a paradigm shift. In the old days, we started with mice first and then went to monkeys. Now, a lot of the thinking is do monkeys first, because you just know much better what kind of doses you end up with and how these doses are actually feasible in the clinic from a safety perspective, sometimes even from a manufacturing perspective, because AAV is very expensive to make and high doses are very expensive. So I think that was a bit of a paradigm shift over the last 10 years or so.
Right.
And what is interesting,
because in some cases, like when you look at mouse transduction efficiency, for instance, into liver being a target organ, for instance, right? Once the serotype of AAV that you use may not have the same transduction efficiency in the mouse and the monkey, and then it's a big question mark as to which one is right to project the human. I think you hit the nail right on the head, which is today we have a bit of a benefit because there has been so much clinical experience that teams can sort of look to see, okay, what was someone else's monkey data with this particular serotype? What was their human data in terms of the level of conservation, let's say, of transduction efficiency that they saw, and help that to guide. But it's was not straightforward in the beginning, particularly because it's hard to know which one's right. Is it the mouse or is it the monkey? You want to think it's the monkey because it might be closer to humans, but that isn't always the case, as we've seen with actually some lipid nanoparticle drugs. So it's complicated.
Right. In addition to that, I mean, the liver is a fairly, I think, comparable organ between smaller animals and large animals, a couple of cell types. But if you look something very complex like the eye, for example, there's a lot of different barriers that are very different in different disease models, and also the size of the eye and so on. There's a lot of additional structural components you need to consider, because these are the structural components the AAV has to overcome sometimes, depending on your route of administration to get in. So even then, you can, certain aspects, you can study them in the mouse or in a rabbit or in a monkey, but it gets complicated very quickly. I think another ethical consideration, of course, is the fact that AAV is immunogenic prevents also some patients from being treated at all. That means you tend to pre-screen basically a patient population for a certain titer of pre-existing antibodies against AAV. And at some point during the trial, you have to make a very hard decision, excluding certain patients based on that titer. That's an unfortunate limitation of the technology right now, but there's a lot of people working on mitigation strategies to get rid of these antibodies before the first treatment, one way or the other. But for the moment, we're still stuck with that, I think.
Exactly. And so, I mean, I think one of the things for me that this always tees up is you've got so many levels where one model organism might be better at predicting than another. But at the end of the day, you have this confusing set of math, right? Because oftentimes your mouse model will be your actual disease model. And that will be the one sort of giving you a guesstimate of, okay, how many cells do I have to correct in order to see the benefit? And then you try to look across to, a non-human primate model and say, okay, now what's the dose that gets me there? Because I think my transduction efficiency is better conserved. Then you have the bigger question of, is the payload, also well conserved between mouse and human? Can I trust the data that I'm seeing in the disease model? There's all these layers, and I guess I always worried, especially at the sort of informed consent stage, right? For subjects who are considering joining a trial, how do we ensure we're sort of properly communicating the science, both to them and to their provider, the PI of the trial, like the caveats that we see this in the mouse, we see this in the NHP. If the NHP is correct, then we think every dose level we've selected is going to have an opportunity for efficacy, but we might be wrong, basically. Because I think that's a position that a lot of teams are often in, especially before the first patient is dosed.
I think many times, make very clear, this is an experimental treatment or therapy. And this is a safety phase 1-2 most of the time. So we really tried here to assess safety. And I think those are very important things to communicate very early through the KOLs a company should work with, because the KOLs know usually, and the clinicians, sometimes they're the same, key opinion leaders and clinicians that know the patient community the best. So they should be able to communicate this well. And then sometimes even working directly with the patient advocacy groups, like not, raise false hopes, but saying this is sort of experimental. Here's the data we have so far. This is what we want to do in the clinic, and make them stakeholder in the whole process.
Yeah.
And I think in terms of then the lowest dose group, which is always an interesting question, I think we had cases where, if it's, for example, a rare pediatric disease with different severity levels, you usually enroll on the lowest dose, like older patients with attenuated disease that, most likely would not benefit anyway, because the disease has manifested itself, unfortunately, so advanced that you cannot reverse it. And I had some amazing cases where, some of these patients came to us and said, please use us for the lowest dose, because I want to do this for the community. So that was quite remarkable.
Yeah.
On the flip side, the regulators have also pushed us even in the past to go a little bit higher from our starting dose. We were very, conservative and on the safe side. And then during the IND discussions or pre-IND discussions, they actually said, we want you to go a little bit higher on your starting dose, because we want to have at least a chance that even those first patients have some limited benefit from your therapy. So it's really a conversation between those three, the regulators, the patient community, and whatever risk the company wants to take on at that early stage. And I think that's sort of that triangle that works pretty well.
Right. And to your point, like it's something where the conversation is going to be different from disease to disease, because sometimes the liabilities of the disease, for instance, if it has a liver presentation, might mean that your tox profile could be different for those patients, as we've seen, right? And so from disease to disease, it may be different from the doses involved. Hopefully, your first dose is within a level that we've explored before in the clinic by now. When we were starting out way back when on the Shire and Sangamo work that we did together, like there wasn't as much experience as we have today. And so there is a lot more experience knowing what may be tolerable and what may be sort of pushing the limits. And to your point, I think having that conversation with the agency and being open to the concept that they may actually ask you to start higher, and that might be the right thing is an important thing to think about.
I'm also fairly optimistic, just to add that on, that we have so many advances right now in AAV engineering that this will allow us to really significantly lower doses in the future to all this sort of off-target problems, either within the liver or outside, will hopefully go away if we are able to lower our doses by 1 or 2 logs. And that helps on the COGS side. I mean, we have like lower cost of goods, but we also have much lower exposure of the AAV at the same efficacy. And there's some really encouraging data for some amazing companies that make a huge difference, and academic groups, of course.
Right. And it's again, it's an important point, right? If we can lower the dose that's required to get the same level of transduction without having to potentially, boost the level of the transgene expression, because we know that itself can also cause problems in certain tissues. It's really still a little bit of artistry to figure out what the right choices are for your product and your patient population. But at this point, we have a lot more data that allows us to have a better sense that, yes, we're balancing that risk benefit for each patient. Why don't we shift gears and talk about an even more complicated topic, if we can, which is off-target and undesirable on-target editing profiles, and a little bit of the science behind that, as well as sort of the ethics of how do you set up a target product profile for therapies in this space. And, I think for me, certainly there have been leaps and bounds in the past decade in terms of our ability to catalog the on and off target editing that is seen from using a gene editing technology, although there's probably still room to grow there. But why don't you take us through sort of a little bit of the science background here, and then we can start to talk about how teams sort of approach this in terms of the ethical considerations.
Yeah, absolutely. So I think for the moment, maybe let's talk about technologies that causing a double-strand break by using a nuclease, like either FOG1 associated with zinc finger or Cas9 or any of the Cas family members. It's important that, these double-strand breaks are, part of life. We basically have double-strand breaks in our genomes at all times. Every day, we have about 10 double-strand breaks, up to 50 actually per cell. And if you take a flight from the East Coast to Europe, you probably add a few more. I think it's about 1 in 100 during that time. And we also sometimes use that technology, for example, in chemotherapy, where we add about 50 double-strand breaks per cell. So it's quite a big load. And we have all these sort of evolved DNA repair systems that are very good at taking care of them. And we need to know them well because we sort of take advantage of them in sort of gene editing that's going through these double-strand breaks. And they're usually repaired pretty faithfully, either by homologous recombination or non-homologous end joining. And one of the things that always bugged me in the gene and cell therapy field is that presenters at these meetings always said, oh, it's an error-prone pathway. And coming from that field, it's pretty clear that if you have a cut that is actually made by nuclease that's actually sticky, your cell is pretty good at basically bringing that together error-free. And there's some really cool studies, I think from Scott Wolf's lab, that showed like about 99% of these ligation steps are error-free, but eventually the nucleus will slip and make a mistake. And these mistakes we can detect, and that's sort of a readout. We call them insertions and deletions or short indels. And that gives the field sort of the opportunity to use this as a proxy for nucleus activity. So we can do this now obviously we can monitor this at the intended sequence, the on target, but we also have now methodology. We can look all over the genome to find off targets that way with a very, sensitive technology, next generation sequencing. And that sort of is the background that we have right now to assess the on and off target activity in cells or even in vivo as well, if we can do actually biopsies.
Right.
And of course, this is something that other modalities cannot do. It always struck me as a bit unfair that, small molecules or biologics, they cannot really assess these off targets as readily, as sensitive as we can. But of course, it's appropriate since these changes are permanent and we need to really exactly know where else do we cut in the genome.
Well, and I think you brought up something important, right? So double-strand breaks happen in your, cells all the time at a certain frequency. Introducing one of these enzymes into a cell increases that frequency, and hopefully focuses at a particular on-target spot that we want to edit, right? However, the risk is that you also get, off-target edits somewhere else in the genome, or that the edit you're trying to generate at the target site is edited in an untoward way, in a way that's not desirable, either because it re-anneals to another part of the genome and causes a translocation or what have you. And you pointed out that we have tools at this point, like next-gen sequencing, that allows us to really survey in a much in a very deep way these edits and what happens. But the limitation of all those technologies is going to be sort of the background rate of sequencing error, if you will, because you can't tell if that's a true edit or noise, right? Because sequencing error also occurs. And so it's impossible to distinguish those. And so on the one hand, we've gotten a lot better at being able to survey very deeply, across the genome for untoward events that we're trying to avoid. But at the same time, there's always going to be a limitation to that. And I think, that's another, I guess, concept that it strikes me as very hard to communicate, even amongst scientists, much less, caregivers, KOLs, patients who are considering these therapies, the unknowns around this. And how have you seen teams try to communicate this, and how do you think we should aim to communicate this so that if someone's volunteering for a trial, they understand?
Absolutely. I think what always helps to communicate this is sort of the ratio between on and off target activity, right? If you establish and that actually works pretty well in vitro. This is something that actually we've seen. If you establish this in an in vitro system, it translates pretty well in vivo as well, in both mice and NHPs. So let's say we have a ratio of like 100, right? For each 10% on-target activity, you have one side that has like a 0.01 percent activity. Then you get very quickly in sort of the detection limit that you mentioned, right? So if it basically your target product profile is like, oh, you need only 5% editing, let's say in a liver cell, then you're probably in the noise of the assay. And that's just something you can do it as hard as you can. What you can do though is you can push systems a little bit, like in your tox studies, you bring it up to like 30, 40%. So you work on like these worst case scenarios, either in vivo, you can also do this in cells. If you talk, for example, about karyotyping or translocations, you can express these nucleases, for example, with AAV over a long time versus, for example, mRNA, which is just a few hours, and really push the systems to see, okay, what's the consequence of these on targets in vitro and also in vivo? For example, one time we got lucky in terms of, and we will talk maybe about in vivo models as well, but we had an on-target and off-target in NHPs that was conserved in the human genome as well. So we were actually able to sort of follow that off-target in vivo over several months. And it was actually similar. We had 9% on-target activity, but we never could actually detect this off-target despite, AAV-mediated long-term exposure to the nuclease. So that was a good way to de-risk this. And this is data we actually presented back at the RAC, so this is all public available information. But it's also something we presented at the IRB, for example. When we made the case, yes, this is the first time we do editing. This is the first time this is done. And here's a safety package and here's our data. And I think that, again, that ratio between on and off target activity is something I'm still seeing in the field being used also in the context of CRISPR. And I think it also informs sort of the FDA guidance on these technologies, how to sort of assess the safety profile of these editors, no matter what the platform is.
Absolutely. And you pointed out something really important is one of the challenges with these technologies is that even if the site you're trying to edit in the gene of interest is conserved across species, your off targets won't be, generally speaking. You guys got lucky with where there was a conserved off target, right? But that's not always the case. And so, to your point, I think what I've seen a lot of teams do, they're triangulating between, human, maybe they start screening in human immortalized cells, but then quickly move to human primary cells to establish the sort of bona fide on and off target ratios that you're talking about and the profile. of where they're seeing the off targets. And then you also look in similar cells from the species of interest that you're going to use in your tox programs to try and see if you see maybe not an exact conserved off-target space, but something else in there that you can use almost as a proxy for that off-target activity. And then you're making an argument at that point of, here's what we see in vitro, here's maybe what we see in an animal model, and here's why we think this is, we've got 1,000-fold enrichment for activity at the on target, or 100-fold, or whatever the case may be. Is that also what you've seen teams do?
No, absolutely. And I think there's a few other considerations. You mentioned primary cells. Of course, they're much better than any cell line. Because in cell lines, you encounter sometimes the problem that your transgene, your on or off target is not even there, or it is there because they have a messed up karyotype, multiple copies, which kind of screws the results. So primary cells help from ideally multiple sources, multiple donors. Of course, then you run into issue. First of all, primary cells are not always easy to be transduced by AAV. And then also there's limited supply because some of them, come from dead donors, and you basically have a limit there, how much you can actually use. I think iPSC primary cells work, but they're not as diverse. And there's the added complication that sometimes some of the nucleus platforms are influenced by epigenomics. That means you have sometimes heterochromatin loci that cannot be easily cut by some of these. So there's lots of variability that you have to take into account if you use multiple primary cells.
And that's a great point, right? It's not just the genetic variability, like the actual sequence of DNA differences from person to person that might impact this, but it's also, chromatin state, patient to patient. If, one locus that's an off target is a little more open in one subject versus another, you might see something very, very different in terms of the rate of off target. So it is, it's complicated.
Absolutely. And I think we talked about the exposure also of the editor. And I think that also plays a little bit into, if you look at the bar for safety bar for different approaches. For example, we talked about germline editing earlier, right? That's probably the highest bar, because any off-target you cause there will be permanently fixed and passed on to future generations. And that was extensively discussed in 2015 at that conference, because that's what people really got into the weeds, how good is good enough for off-targets. Then you have a group maybe of diseases where maybe even age-related diseases like ocular diseases, like geographic atrophy, where your follow-up is not that long, right? So there's not much time for the off-targets to really do much damage, even if you have them. That's why the bar is a little bit lower. But then you have something like devastating rare pediatric diseases where you really want to have an intervention very quickly, and you don't want to necessarily screen for like the next 5 years for the absolute perfect editor. So there, I think an intervention is much more urgent. And I think there the bar is a little bit lower.
And it's also the sort of ex vivo versus in vivo as well, like in some regards, right? I think, with the ex vivo, again, maybe you're doing a pulse of the editor to the cells, so it's super low activity or attenuated time of activity. And then the hope is that reduces the sort of off-target potential versus when you're, supplying directly in vivo the editor by, for instance, something that's long-lived like an AAV. That's much more complicated and makes doing this math a little more difficult in my mind, at least. I don't know how you think about it.
No, absolutely. And I think it also matters, of course, in addition to that, where the off-targets are. I mean, a lot of work should go into what's the exact loci. Is it a vast intergenic region, or is it something that's on your oncogene tumor suppressor list? Because then you definitely don't want that. And that's probably like an exclusion criteria for that particular lead. And you mentioned translocations. There are potential translocations between the on and the off targets, and also amongst the off targets. But you can model all this first in silico, and then you can also experimentally detect these potential events, which in the worst case scenario could be an oncogenic fusion protein, right? But you can sort of de-risk this to a certain degree and sort of say, can I model this in vitro? Again, have long exposure, worst case scenario, and see what the consequences are of these off targets. And if you don't find anything in these really sort of when you push the system in these experiments, I think you have a pretty good argument with the FDA and also the patients that this is fairly safe.
Yeah. And I think the other thing you mentioned earlier that resonated with me is the indication that you're trying to treat matters, right? In terms of how you think about this. If this is something that is affecting, let's say, for the sake of argument, a 90-year-old person who may not have 5 decades ahead of them of life, like that for me is a different calculus of the you still want to aim for the cleanest possible enzyme or guide or what have you, but at the same time, there's a desire to get into the clinic expediently, right? To start to see if we can help those patients. On the flip side, if this is to your point, a pediatric disease, I think it should and ought give teams pause to think about how am I approaching this? What is the right target product profile when I think about the lifetime of this patient, not just their initial treatment here.
Yeah, absolutely. And I think all that is contributing to your discussion. Yeah.
Yeah. So I think we're nearing the end of our time. And so I get to ask you my favorite question. And what's fun about this one is I think I'm gonna I have a personal different answer, and maybe maybe it's similar to yours, but than I usually do, just based on the topics we've just discussed. So if you could wave a wand and fix one thing for cell and gene therapy development, what would it be?
I think technically it's really delivery, right? We want to improve our delivery methods to the point where we can repeat dose ideally to effect, meaning to our intended threshold of on-target activity. And tied to that is also the affordability, to really have something that way we don't have to make huge bioreactors with AAV, which is very expensive and very involved on the CMC side, something that really improves access for all patients. So a delivery method that maybe can also sidestep pre-existing antibodies even better. So basically a delivery method that benefits as many patients as possible. That's my...
Absolutely. And so I think that's very similar to my answer. It's basically the ability to redose, to dose to effect, so if a certain dose level, works for 90% of patients, but not 100%, being able to, dose that 10% additionally if needed. And I would also maybe add in there, since we're dreaming here, the ability for our favorite preclinical models to actually model the transduction or transfection well, so that we know that the doses we're projecting, even in that first dose group, to go back to our start of our conversation, might have a chance of having the benefit for the patients.
Right. And just to add, I think on the sort of my ethical side, but also industry-wide side, I think I really wish we had more sort of larger companies taking interest again in gene cell therapy. There was a lot of opting out lately, because I think this company is so important for the later stages. Clinical deployment is extremely complex and expensive. That's what they're very good at. And I think, while small startups are very good at the early stages, I think we really need to work together and play to the strengths of each different size of biotech. And hopefully we get a little bit more involvement there in the future again.
Absolutely. I would love to see that as well. So Thomas Wechsler, thank you so much for spending time with us today. We really appreciate your insight and the conversation here. And I know we're going to be talking about ethics and more on this podcast, so I'm very excited for that. And perhaps we'll have you on again for some of those conversations as well.
Thank you so much. It was a pleasure.